separate, we each have our own teams that we're working with. So Danil Naji is working with David. Dan Taeyang and Annalie Berner, I don't know if she's here tonight. She's also on our team. And so here's how the order is going to go tonight. So the first presentation is by Ali Brivanlou, who's a biologist, computational biologist. Then we're Lars Dietrich, who's an assistant professor here at Columbia, also computational biologist, microbiology. Eitan Grinspun in the computer science department. And those three people are working with David Benjamin on his project. Then we have Clay Lacefield from the neuroscience department at Columbia. Scott Klein, a journalist, works at ProPublica. Alex Tyrell from Thompson Reuters. And Dennis Tenen from the English department. And Mark Hansen, where is he? Statistician and director of the Brown Center for Media Innovation in the journalism school. So each person is going to speak for 5 minutes. We're going to hold up a timer. Sometimes a speaker is going to be amended and with an extra video or animation, something like that. And then afterwards, we're going to have a discussion. And the idea of the grant as a whole is to foster collaboration around the ways in which data is used in the university, because data means very different things in a lot of different disciplines. But as a project that's run in the architecture school, what we're trying to do is collaborate with journalists or with scientists to think about how to either in the case of journalism, to help tell stories with data in ways they might not have done before, and in the case of science, to communicate very complex computational ideas to a more general public. And so in each case, data means something very different. And so we're very happy that today the conversation is going to span from the humanities to the sciences and beyond. And that's very important for this project, that it doesn't only stay in the realm of data is not only about science. Okay, so Ali Brivanlou is going to start. Yeah. Oh, David's got an introduction. Yeah.
Great, thanks, Laura. And I just want to say a few words by way of introduction, and then we'll launch right into the presentations. So I have a video up there that we can play, ideally. And first I should just thank again, as Laura already has, Thomson Reuters for their very generous support for this project. They've basically made this whole thing happen. Sorry, I'll need to start the video, so if you can stop that. Great. Stop. Okay. Great. So can you guys restart this in QuickTime and bring it to the beginning while I just say a few more words? Okay, great. Thanks. So I wanted to just really quickly reiterate the mission of this project. This project started about 1.5 years ago, and the original mission were these words, which I still think are really interesting and important. The idea was, in quotes, to redefine the state of the art in dynamically visualizing existing, evolving, and future data sets. So in my mind, this is not only about like the current version of data visualization, but also an expanded field of possibilities for data visualization. We've been really excited to collaborate with a range of kind of top university researchers here at Columbia and beyond to explore new ways in which data visualization will impact what we think are some of the most important questions of our time in many fields. And we think that this explosion of data has led to great opportunities for this expanded version of what data visualization is. So tonight you'll hear these several examples. I won't try to summarize any of them, but I did want to just give a flavor for the way that we might think about data visualization at the School of Architecture, because I believe that this explosion of data has impacted many fields, including architecture. We have our own version of it. Of course, we know that there's been this kind of quiet yet revolutionary transition from analog tools to digital tools in architecture. We know that parametric modeling and BIM, building information management or building information modeling, and digital simulation are increasingly an important part of every complex building project. We know that the relationships between inputs and outputs and the number of design iterations and the complexities of most projects are beyond what a single human brain can process alone. So we're obviously relying on computers for a lot of this. And at the same time, we may rightfully be cautious because we know that data can be used for some suspicious ends like metric-driven design, value engineering, and cold-blooded efficiency. But I would like to argue, just as part of the context of what we're going to do with data in architecture and beyond, that data can also be used for enhancing our creativity, for exploring wide design spaces, for reducing our human blind spots and overcoming our kind of narrow human assumptions, for discovering new possibilities and new high-performing but creative design options that would not occur to us otherwise, for even things like negotiating between competing objectives. So not delivering one single optimized result, but allowing and actually demanding for a discussion and a debate about values, about what our priorities are for architecture, about how to gain a little bit of one metric while losing a little bit of another, or vice versa, and which is most important. So to me, data visualization involves art as well as science. It involves creativity as well as efficiency. Data visualization is an important ingredient in the future of architectural design. And therefore, of course, data visualization is an important ingredient in architectural education. So along these lines, I believe we have much to learn from the examples of other fields. I think it will be an interesting night for us as architects, those of us in the room who are architects, to look to other fields for examples that might be helpful and interesting to us, to learn from these fields, and then to reflect back on our own field of architecture and the way that data and data visualization might impact it. So thank you for that. And with that, I would like to introduce the first speaker. We'll just give very brief introductions, as Laura has already mentioned. Ali Brivanlou, who is a professor at the school, actually, but also is the Robert and Harriet Helbrun Professor of Embryology and Stem Cell Biology at Rockefeller University. And it's a great pleasure to have you here, Ali.
Thank you, David, for this great introduction. And I want to second the idea that architecture and science in both directions can beneficiate from each other, not only in terms of experience, technology, and approach and design, but also in terms of learning and moving forward. I have a movie that hopefully will be ready to be launched soon, and I'm going to try to stay within my 5 minutes. As David said, I am a professor of embryology and stem cell biology at the Rockefeller University. I've been there for 20 years. And last year I had the honors of teaching a class with David Benjamin here in regenerative architecture. And from that I learned a lot. I learned first that the two sides of Manhattan can contribute toward the same projects. But this kind of output in terms of data, and you heard that data has many different meanings, is what has preoccupied me for most of my professional life. You just saw a fertilized egg become a tadpole. You're seeing it again in a very fast, dynamic motion. On the other side of the screen, you're looking similar things happening with the fish, and now, finally, our own. This is a human development. And from this, you have already seen that there is tremendous dynamic, and it stands in contrast with human designs of buildings and urbanism. A biological system such as the frog develops very fast. It changes shape, it changes contour, it adapts to its environment. And of course, this is the product of millions of years of evolution in order to be able to handle all the environmental changes that a tadpole has to go through, and everything else that follows in terms of being independent in where you live. We had students last year coming to the lab, and we built special cameras for them to actually image the same tadpole from 4 different angles. We're moving now to 8 different angles. That gives us the kind of single cell resolution that would allow us to map this thing in time and in space. And architecture students bring their own innovation to this approach. They actually take a sphere and flatten it, so you can look at movements of individual cells two-dimensionally rather than going to a sphere. These are all change of shapes and contours that nature has used over hundreds of millions of years to generate entities that are extremely adaptive. We can play with these systems. For example, we know enough about the way a tadpole makes his head and his tail to change the morphology, the natural morphology of a frog. We can make, for example, tadpoles with 2 heads or 5 tails and 6 eyes. This is the way embryologists and biologists show that they understand something. They actually take it apart, they deconstruct it, and then they reconstruct it back to show that it works. And that gives us the proof of understanding. There's a huge code to be cracked that is shown in that diagram, and this is post-genomic code. It's no longer about genes and sequences of DNA that we already know and we have in the pocket, in our back pocket, but it's about the way this thing works to generate life and adaptation. So the essence of cell signaling, basically, in early development is what determines shape. And as I said, this shape changes very dynamically. We can understand how these shapes are made by using a whole bunch of microfluidic robots and other sophisticated techniques. But at the end of the day, what we really want to know is the instructive signals, not the permissive ones that are involved in making a tadpole look like a tadpole. And as you saw from the beginning of the first movie, that program is very robust. So when you align 10 eggs together, they will all develop the same shape tadpole with the same space of time. We can manipulate the information that every single cell receives in a tadpole and generate shapes that do not exist. For example, a tadpole with 2 heads. And this tadpole lives happily for a long period of time, and it tells us where the information for generating a second brain or eyes come from. We can recapitulate this in the human embryo, shown here on the left side of the screen, because evolution keeps things very constant. We can also look at data 4-dimensionally. This is a world record imaging of a tadpole. It's about 1 millimeter, and you're looking at a Z-stack in a vertical plane in a 30 confocal microscope. So the resolution is very high. We can generate, based on this knowledge, organisms that are chimeras. These 2 are mouse and human chimeras. Human cells are in red, mouse cells are in green. In the previous one, the human eyes were in blue and the mouse was in white. This again tells you that we can make shapes in nature compatible to one another. So starting with this kind of observation and this kind of information, we arm our students, David and I here at the architecture school, not necessarily with the knowledge of becoming a scientist, but basically with the essentials of what does it take, or what are the basic rules of generating shapes and form. These are examples of students, one from last year and several from this year. The size of the class has increased. And this is their contribution to the ideas. After you explain what's going on, they come up with a generation of materials, for example, that have dynamic properties, thinking about the way those materials can be assembled to allow, all based on biological principles, the way architecture can be modifying its design. So the logic of cell growth, differentiation, and morphogenesis can be a template for dynamic architecture. And as you can see, the students are expressing that extremely well. A design ecosystem where humans and biological cells cooperate to generate a different form where neither human alone or nature alone can generate, I think is the next frontier for dynamic architecture. And the ideas are as crazy as you can imagine them to be, like this one, which basically is a floating roof and a part of an urban structure. This might, of course, involve the design of new material to make buildings basically come alive, or the concept of living buildings. They may offer some of the dynamic feature of natural living systems, including growth, repair, and adaptation. And I think, to me, that would be the way we can coalesce our knowledge in science with the knowledge in architectural designs. Applications may involve the design and execution of new biologically based building systems. And these systems may be more complex than the materials currently produced in any lab, including my own lab. And the process may be in the form of organisms that act as a factory to produce building material on site. Other examples may include bioplastics that do not require petroleum, bacteria that fuse sand into brick without baking, wood that is extremely strong, flexible, and durable, biosensors that change color when they detect toxins or structural problems, microbes that find and heal cracks in concrete, or termites that eat construction waste, as well as old cars and cell phones. These new bio-derived materials may offer high performance, lower cost, lower carbon emission, and less waste than the natural counterpart. Designing with biology is a logical and inevitable turn in architectural creation, as far as I'm concerned, and designing with biology is what we do. I thank you very much for your attention, and I think we're going to take questions at the end, or right now? Okay, perfect. Thank you.
Thanks a lot, Ali. And so our next speaker is Lars Dietrich, assistant professor in biology at Columbia.
Okay, hello everybody. So it's actually great to follow up Ali's talk, because what really drives me to biology is the enormous diversity of shapes and patterns that we can find in nature. And that's really what I ultimately try to understand. And like he also pointed out, one of the building blocks are single cells that then come together to form multicellular communities that form all this diversity. And so how do we get to this diversity, although it's always single cells that are the building blocks? And the other weird thing is that I'm actually a microbiologist. And you typically, when you think about microbiology, you think about bacteria, and bacteria more associated with something like this, which is disease. And one of the things that I want to get across today to you is that actually only a very tiny fraction of bacteria actually cause disease. And even those bad bacteria can create really fascinating shapes, such as this one right here. And this is actually a bacterial colony growing on the surface of an agar plate and stained with a red dye. This particular structure is made by the bacterium Pseudomonas aeruginosa. And there's a lot to say about this bacterium, but I just want to highlight one thing, and that's actually that it's the most common organism in holy water, if you go to church. But it's also important to point out that they can come together to form these fascinating structures. And they are about the size of 1/4, and within this structure there are 5 billion cells. And the question is, how do they manage to all orchestrate themselves to communicate with each other to form this structure? So we want to understand how to get from the single cell to this multicellular community. And one aspect that's actually very important in terms of disease is that once they form these structures, they're 1,000 times more resistant to antibiotics than when they form just single cells. So in very many disease states, they actually form these communities. We need to understand how they form them in order to form new drugs. So an important aspect here is that everything is driven ultimately by gene expression, what kind of genes are being switched on and off. And each of these bacteria, they have the same genome. It's a circular genome. It's shown here. This genome, each of these lines is one gene, and they have about 6,000 genes. And you can imagine it like a necklace with pearls, each pearl representing one gene, and they have all the same set of genes, as I mentioned. What's now really important is the pattern by which these genes are switched on or off. So for example, it could be these ones, but it's not enough. It's also about how the gene products ultimately interact with each other in different ways. And this will ultimately tell you what the cell is actually doing at the end. So depending on what kind of pattern of gene expression you have, they will do different things and create different types of communities. And we look at these communities with a high-resolution digital microscope on these colonies that are grown on agar plates. Here we can see them close up. And then we just monitor them. And if you look at a reference strain or wild type, that's how it looks like. It's smooth and pretty boring, actually. And this is how it looks like after 3 days. Now we make a random mutant, for example, right here. This will affect this network, and then something amazing happens. the development of this colony completely changes. It goes from this smooth to a spread colony, and it forms this concentric rings in the center and this more radial spokes toward the outside by just making one mutant. So you get a better feel for these wrinkles. We can actually get 3D images of those. So here you see them from the top. We can rotate them, and we can actually in real time determine surface to volume ratios and without fixing anything. And I should say that they are not hollow here. This is actually filled and packed with cells. It's just this type of microscopy only allows us to look at their topology. The reason why they actually wrinkle is we think they want to increase the surface to volume ratio, because these type of bacteria actually need access to oxygen. Like us, they really like oxygen. They need it to breathe. And it's similar, for example, to our lungs or roots. They increase the surface to volume ratio to get more oxygen under certain conditions. So, and each cell can actually monitor how much oxygen is around. And they have sensors that can sense the amount of oxygen. This leads into a signal transduction pathway, which affects gene expression patterns. And each cell is doing that. And then as a community, they come together to form these structured communities. So the idea was now to understand what are the genes that are being involved. And for this, we do a screen where we basically mutate every single gene in the genome and then go step by step. So now it's very slow, sorry. We'll go through this. Almost done. Okay, and then we basically spot them on the agar plates, and we look at all the different morphologies based on these mutants. And I don't want to do this again. And we basically screened 6,000 genes, and it turns out that 1,000 of them are essential, so you can actually, if you mutate them, the cells just die. But we can look at 5,000 different mutants. And we follow them over 5 days, and this gives us 25,000 images. And this in itself is not a very big number, but for us as a lab, it's already quite substantial. And there are two problems, as David also pointed out. The first problem is storage of all this data, and the other problem is analysis. And the way we want to analyze them is based on their morphologies. And we find different morphotypes. These are extreme cases. And you can very easily see that they are very different. So each of them is just a single gene mutation. And now what the idea is that we find different genes that have nothing, that we didn't know that they were doing anything together, and see that they have the same morphology. So we can basically group them based on the morphotype and then go back to the genotype and classify genes based on that. And here again, you see the genome of the Pseudomonas genome, and in the background, the gray is all individual genes, and the colored ones are the genes that we found to have a wrinkled morphology. And we found 145 wrinkled mutants. And that's where Daniel and David came in. What they helped us to do is to look at all of them and extract information. And we applied two methods. The first was that we basically told them what we are interested in. For example, in these spokes that you see, these radial spokes that they form, they helped us to identify those very quickly. And with this first method, the goal was basically to speed up what we anyway would do as humans. We would look at those images and classify them accordingly. And then they extracted different parts of information very fast. The other one is actually more intriguing to me, is that they look for features that we as humans wouldn't really recognize. They use different algorithms and just random on our images. And so if there's anything that you can categorize. And one way you can also do this is by, for example, applying face recognition software on the morphologies and thereby classify them, where you then find different components and do all this analysis and put them together. and I'm going just very quickly through this. And the other aspect is to also apply this to a moving image. So not just to still images, but also to moving images. And applying different algorithms and methods to analyze them. And with having the movement in addition as a criterion, we hope to find even more underlying principles of how these structures forms. Here, for example, in the next one, we look at how much surface there is to wrinkle surface, and it's very quickly, and here's a network analysis of how these wrinkles form. And then once we extract all this information, we use machine learning methods to basically analyze these structures. And there are two ways again. One where we basically tell them what we want to find and we adjust it according to that, and one where it's just running on its own, independent. And just to finish this up, so what we basically start out with is this data set and then get this analysis. And from the analysis, we learn more about the biology, which allows us to generate more data. We can also use this analysis to do modeling. So we actually have a collaboration with biomathematicians that can use all this data to model the structures. And it can also, again, help us to do better analysis. And the third aspect that I'm pretty excited about is to make all this data available to the scientific community. And here, Daniel and Damon, they actually, oh, it's too bad. So basically, they created a website where you can look at all this data on a platform and quickly you look for one gene that you're interested in, and it will show you all the other genes that show very similar morphologies. And this will be made available to not only to the Monas community, but in general to all microbiologists. And we want to make this available within a year or so. And with this, I want to thank you.
Okay, the next speaker is Mark Hansen. He's in the journalism school and does many projects with us.
Thanks, Laura. And I apologize, I guess the complexity. Well, anyway, I'm not going to show any graphics today, although I do feel a lot like one of the 145 wrinkled mutants. So my background is... A couple people got that. Anyway, so my background is in statistics. I earned a PhD from Berkeley about 1 million years ago. And in the intervening years, I've been fortunate enough to work with a number of artists and architects. I've collaborated on pieces that hang in the lobby of the New York Times building, the Public Theater, and soon a piece for the 9/11 Memorial Museum. But it's my great pleasure this evening to talk about the work I've done with Laura and the Spatial Information Design Lab in the last year and a half. Actually, I met Laura first during a project. Actually, I met you even prior to that, but our first significant project was work with Diller, Scofidio, and Renfro under the watchful eye of Paul Virilio. The piece was called Terre Natale, and it was a data-heavy project that dealt with human migration writ large. It was a large circular projection that was divided into a series of theatrical scenes. Each had its own logic, and each had to do with why we move, why we cross borders. Is it to take a job? In a scene about remittances and how money comes home? Is it because of political reasons? In a scene about political refugees? Is it because of the effects of climate change and the controversial term environmental refugees? And so the piece itself dealt with a series of these scenes, and like I said, had a number of data sets as curated by the UN and other independent organizations. And if any of you have had the experience of trying to slam multiple data sets together, it can be a joy. Laura and I then collaborated on a course in the fall of last year. I joined Columbia a year and a half ago. In the fall of last year, we taught a course that had an equal number of architects and journalists. It was a seminar. Our starting point was that both architects and journalists take the city or some aspect of the city as their beats, and yet each study the city in very different ways. The class was an incredible learning experience for me personally, comparing narrative, different forms of narrative, the built and the written. I know we got a lot out of the class. I don't know how much the students got out of the class, but I certainly learned a great deal. And maybe ultimately that's why we teach classes, because we learn a great deal. Currently, Laura in the Spatial Information Design Lab and a team of journalists and I are working on a design for the lobby of the Mind-Brain Behavior Institute. This is the first building that's going up in the Manhattanville campus. Brain science, like the scientific examples we've seen so far, ride on imagery, rich, sometimes complex, sometimes very complicated, sometimes misunderstood imagery. The metaphor for the MBBI, Mind-Brain Behavior Institute piece, is projection, data projection. So building on the notion of the grand tour, which is a tool for exploring high-dimensional spaces through a series of animated two-dimensional projections. You can think of it as taking a series of sort of rotating a data cloud around, but instead of having the cloud living in 3 space, it lives in 20 or 100 or 1,000 space. So it builds on that idea and divides a huge lobby space into several areas, each projecting, if you will, the data of neuroscience into different viewpoints. So a scientific paper, for example, lives in the context of other papers, is cited or ignored. The work was funded by one or more sources. The labs involved have a history of collaborating or competing with other labs, all producing a kind of rich set of networks that can be displayed or projected. An individual artifact, like a paper, can be projected into that space. In another region of the lobby, we put the research in the context of the physical brain itself, or perhaps a neuron. So in the case, you could imagine sort of old school functional MRI that binds locations in the brain with certain kinds of behaviors or certain kinds of illnesses. This idea is taken to an extreme in what we're calling the brain index, where individual artifacts of research will be displayed or made displayed relative to the background of a large brain or the geography of a brain or an individual neuron. Just to give you a sense of the scale, from 7 feet up to 27 feet will be a large projection space, and then we've imagined at least large moving LED screens that are at human scale that can take data from a particular location in the brain and bring it down sort of to a human scale. So whether we're projecting science onto the press and social media, or the workplace taking place in the building, or spots on the brain, or within networks of papers, our complex sets of data, papers, 3D models, data sets, computational models, are all represented richly in this MBBI lobby space. So I thought I'd end with maybe just saying something about my role in the School of Journalism. We're a little behind architecture in the sense that we're starting to take up computation seriously. The sort of computational acumen of the people in this room is much higher than what you'd find, I think, with a group of journalists. It's probably actually much higher than you'd find with a group of statisticians who are retread mathematicians who are often more interested in deltas and epsilons than anything else, but that's a whole other lifetime ago. That said, data code and algorithms are changing systems of power in our society, and journalists need to be not only conversant with data and data technologies, but also need to be creative in that area. So toward that end, my teaching involves bringing ideas from computer and data science to the journalists, teaching these ideas in a journalistic context. One of the projects from my class, for example, turning up in the work that Scott will talk about, of course, ProPublica added a huge amount to it, but a kernel of it started with Laura and a couple of my students. And in the J School, we started adding new classes to the curriculum, underscoring the importance of knowing data, code, and algorithm as tools for finding and telling stories. So that's my 5 minutes. Thank you.
Thanks, Mark. So our next speaker is Eitan Grinspun. He is Associate Professor of Computer Science at Columbia.
Great. Let me turn off Lux. Okay, thank you so much. Can everybody hear me? So this is detective hairdo. This is a little funny intermission in the middle of our set here. This is joint work with researchers at Adobe, Disney, Weta Digital, and yes, there are some people from Columbia on the list as well. The goal is to avoid days with a bad hairdo, all right? Or more seriously, the goal is to understand what drives simple phenomena like the way our hair sits. How do you get the difference between this hairdo and that hairdo? Yes, that's real hair. And it's inspired in part by the Ig Nobel Award of 2012. Here's the mascot for the Ig Nobel Awards. It's called the Stinker. And the Ig Nobel Awards in physics are given for physics problems that make you laugh first and think second. And researchers came up with the equation that describes the shape of the ponytail. All right? So there's a bunch of physicists and computer scientists that are interested in describing very simple everyday phenomena that surprisingly are still not understood today. And that's where this project came from. This project was also inspired by my own group's background in computer graphics and the work that happens every day in the film industry. When artists are animating and telling stories, they are supported by the underlying physics. So while the story is going on and all of the music goes on and the characters are, and sometimes the physics is not so real. But at any rate, when the artists are being trained, they have to simultaneously tell their story and support the physics. So here we see different examples of one of the famous exercises that's done to this date. It's called the sack of flour exercise. And we see these sacks of flowers. They're simultaneously eager, happy, sad, sleepy, always maintaining their sack of floweriness, so to speak. So when artists are doing this, they have to think about lots of things. They have to think about gravity. They have to think about all of the forces in the physical world, including elasticity, contact, and friction, and use all of those to support the underlying storytelling. And computers come into play today in movies that are doing these animations. So in our group, we developed a model for flexible strands. This model has been used by doctors who are trying to understand catheter insertion during surgery, and it's also been used in Adobe Photoshop to simulate realistic paintbrushes. But in the context of today's discussion, the point is that this same model can model flexible strands such as hair. It can also, the model takes into account several things, including the ability of an elastic material to twist. And as it twists, it assumes certain formations. And those formations, for example, can be a double helix, as you see in biological structures. This is known as a plectoneme. And contact of the rod with itself, of the hair with itself, is vital to forming these kinds of structures. So one of the film studios, Weta Digital, caught on to the fact that we were working on this, and they said, let's take this for a tryout and see if it can serve as a hair model. So here they are testing the strand model and putting it through an obstacle course. And this is from a few years ago. And it looked relatively promising, but there were a lot of challenges to handling massive amounts of hair. So at first we did some animations with them which had about 1,000 hairs. Here it looks like a lot more than 1,000 hairs, but that's because each hair is rendered rather thickly, and each hair is, in a sense, multiplied after the fact. So it's rather coarse simulations. These kind of simulations have already made it into some films. For example, if you watch The Hobbit, you'll see our hair simulations in them on the various characters, but in particular on the animals, like the rabbits here, or these little animals that are being played with here. But notice that these hairs are relatively short. As new movies are being done, such as Rise of the Planet of the Apes and other Hobbit movies, we're looking to really handle massive amounts of long hair. And for that, we're looking at these kind of obstacle courses. We've designed this little car wash for hair, and there's some hair coming through it, and it's getting all bunched up together and braided up. And the idea is to be able to simulate and handle these kind of massive assemblies of complex fibers in a computationally efficient manner. Here is our attempt at trying to comb this mess of hair. And one of the things we've been discovering is that getting the contact right, capturing all the collisions between hairs is vital to getting the types of hairdos we see in everyday life, and in fact to understanding bad hair. So here are some simulations we've done in the past few weeks where we're capturing interactions between hair, and you can see a lot of locking and dreading that's going on, not because of any chemicals or adhesives, just because of the collisions between the hair causing locking. And as we ramp up, let me go back through this. As we ramp up the number of hairs here, you're going to see more and more of these formations happening. So while we watch that, let me tell you some of the things we're struggling with and where we're working with David and Daniil on trying to understand how to improve this technology. One of the things that's happening is that as we move to higher and higher numbers of hairs, so here you see 16,000 hairs, the computation slows down because you get bigger and bigger clumps of hair or islands of hair that are all interacting with each collision affecting the other. Now you really want this because you start to see larger and larger structures such as you get if you don't shampoo your hair for weeks. I don't recommend it. On the other hand, these computations start to go from taking just a few days to taking a week or weeks or months to get a single shot done, which is not feasible in a studio. It's not feasible for L'Oreal when they're trying to understand the different kind of chemicals that can be put into a hair. And so we're looking for efficient algorithms for resolving these. And we're working with Daniil on identifying different kinds of patterns that may happen during these collisions. And we use different visualizations. Here on the left are the velocities of the hair. So we just comb all the hairs out, and even though in practice the hairs are moving in all directions, Daniil has frozen them on the left and is showing their velocities color-coded. And on the right, we're seeing all of the collisions that are happening between the hairs, color-coded by magnitude. And we're looking also at the connectivity between hairs, which hairs are colliding with which other hairs on the scalp. And we're trying to understand what sort of patterns can we extract from this, what can we understand in order to make more efficient algorithms for hair simulation. Thanks very much.
Okay, so I'm introducing Clay Lacefield. As Mark said, we're collaborating with the Mind Brain Behavior Institute, and I think it was just 10 days ago we met Clay as we were walking through one of the labs and were describing kinds of things we did, and he was, well, why don't we try and build a 3D neuron? And Madiha Merchant, who's sitting in the audience there, has been working very hard with Clay and going back and forth between models. And I think we have one of the very few 3D printed neurons in the world. I could claim that, I think. Yeah? Okay.
Hi, my name is Clay Lacefield. I'm a postdoctoral fellow in neuroscience at in the medical center. And like Laura was saying, this is a very new thing that we're starting, a new collaboration. And I've always been interested in trying to communicate the beauty and order of the architecture of neural systems. And it's something that's kind of hard to comprehend because it is so amazingly complex. And so anyway, one of the ways that we decided we could possibly do this is by actually making physical models of some of the neural structures that we see in the study of neuroscience. And so as just a little bit of history, people have known for a long time that this mush up in our heads was important for something. Of course, for a long time, they thought that it was the fluid-filled vessels that we know as the ventricles that were actually doing the important thing in the brain. But around 100 or maybe 150 years ago, a Spaniard named Santiago Ramon y Cajal developed a technique or modified a technique that was able to visualize the precise neural structures of some of the many complex neural networks that form the mammalian brain. from just observing and drawing actually these, some of these neural networks that were sparsely labeled in the mush of the brain, he was able to intuit some very basic principles of neural architecture, such as the neuron doctrine, which is that the brain consists of a lot of cells called neurons, and other things about neural architecture, such that these neurons form connections called synapses. And he found all of this out basically just by looking at the stained tissue. So one of the problems that I see is that what most people think of as neurons, and we can see from our journal of record, Wikipedia, that this is the view that most people have of neurons. And so I thought in order to try to give people an intuitive understanding of some of the beauty and order behind the neural architecture, which underlies our basic perceptions and actions, that we could give physical representations of these of components of our neural structures in our brain. So and this is made possible basically by we have a wealth in the course of doing neuroscience and studying the neural structures, we end up getting lots of 3D models of neurons. And now that 3D printing is relatively high resolution and relatively cheap, then we can actually start to maybe try to embody what's going, the neurons in our brain in this manner. And this might help people in general, and as Laura was talking about with the new ZOMBI, the Zuckerman Mind-Brain Behavior Institute, kind of an unfortunate acronym, is to, the entryway, give people a sense of what's going on in their brains. So, so just to go 100 years after Cajal, currently, we have a number of ways of extracting, three-dimensional models of neurons in the brain. And I'm just going to run through those for a second. One of them is neurolusida reconstructions, which produce these kind of idealistic computer models. The second is using confocal microscopy, and as I'll show you, that's what we ended up settling on for the model that Mediha and I produced today, actually, and serial section electron microscopy. So, so the first that I'll talk about is the neurolusida reconstruction. So this is similar to camera lucida drawings that people used to do, where they would kind of like project a microscope image on top of paper and trace out what they saw. And a lot of the early to mid biological drawings were produced by this method of being able to project the microscope images onto paper and draw them. But now what we can do is we can do this in a microscope where we can actually see how far we're focusing, and from this we can extract three-dimensionality and complete 3D neuron structures. But it's very laborious. It's gives the entire neuron in cellular detail, but it's rather laborious. It takes a long time to do this. The next one that I wanted to talk about was serial section electron microscopy. Even more detail in that you actually use electron microscopes to trace every little dendrite. And in this case, you see the dendritic spines where synapses between neurons are formed in a neuron, and how that's reconstructed into a three-dimensional shape. But it has the finest detail, but it's the most laborious, and it takes a lot of electron microscopy, a lot of processing power, and only reconstructs a very small part of a neuron. So we decided in the past 10 days that we've been doing this to use confocal microscopy, also incidentally first conceptualized by Marvin Minsky, who also came up with artificial intelligence. That's just a random thing. But so it gives great detail, relatively easy to obtain. And so this is a single neuron filled by one of my colleagues in lab from the mouse brain, from the cortex of the mouse brain. So we decided we would try this technique. So this is hopefully a movie of a confocal Z-scan containing this neuron. And as you can see, what confocal microscopy does is, in order to limit the light coming back from a sample, there's a pinhole where all of the light has to come back through this pinhole, and only light coming through this one Z-plane in this microscope slide is able to get through this pinhole. And therefore, as you can see in this in that picture, that you can actually scan down through this neuron and extract three-dimensionality. So after doing this, you can reconstruct this into a three-dimensional model. And so this is the neuron from this slice of mouse brain. It's a 50-micron slice, about 120 microns on either side. So you see that from the last stack, or Z-stack as we call them, of this neuron, we can extract this three-dimensionality and see the kind of the beautiful detail of this neuron, even down to the level of single dendritic spines. And you can see how complex these neurons are. So, so where we are now, this is actually should be updated, but let's see this. Okay, that's not playing. But Medea Merchant from GSAPP has worked with me for these last 2 weeks very closely, and she did some nice editing of these three-dimensional surface maps. And after doing that, we were able to print one of the, I guess, the second 3D printed neuron, right? So this is the same neuron I was showing you. So I think that we're going to try to do this in a larger scale structure, and as you can see that the neural networks are extremely complex. So if you could kind of represent this in in a very physical, tangible kind of way, then maybe you can communicate some very basic things about these, the architecture of these circuits that underlie these very basic things about our nature. So, so anyway, I just wanted to have a few acknowledgments of the head of my lab, Randy Bruno, up at the medical center, some of my colleagues, Amanda Kanischka and Carl Schoonover, who produced who filled some of these single neurons. And I'd like to thank people at GSAPP for supporting this idea and making it happen, such as Laura Kurgan, Jochen Hartmann, and especially Medea Merchant, who's done an amazing amount of work with the reconstructions. And also, I've just signed on a collaborator who has an endless supply of these three-dimensional models so that we can maybe try to reconstruct entire neural circuits using these same methods. So thank you, guys. Anyway, you can come see the neuron later if you want to up there.
So next up is Scott Klein, who, like me, is hard to define exactly what it does, but he's a journalist and has also introduced data journalism to the journalism world, and actually works at ProPublica, which I think is safe to say is one of the best remaining investigative journalism websites that are still around.
Thanks, Laura. Thanks to everybody at Columbia for inviting me. They always put me after the guy with hair models and 3D printed neurons. So I have a bit of a different thing to show you, perhaps another intermission. But as Laura said, I work at ProPublica. ProPublica is the people sort of show of hands. How much kind of deep do I need to go in? Nobody. Good. So that's good. So ProPublica is a nonprofit journalism outlet here in New York City. We're a nonprofit, so we take charitable donations from individuals, from high net worth individuals, and from large grant-making institutions to recapture one of the things that we felt in 2008 was being lost in the destruction of the newspaper industry, which was investigative journalism. I think we all started there thinking we were going to be doing traditional long-form investigative journalism, and I represent somewhat of an outside piece of that, which is trying to rethink computational journalism, data journalism, and especially how we can tell stories using this data in a way that people, regular people, can come and understand the data and how it relates to them in the real world. So we, as I say, we are kind of both engineers and journalists, and we think about things as engineers and as journalists, and we look at systems and wonder how we can kind of deconstruct them, reverse engineer them, and then explain them to an audience who may not know anything about them. We were fascinated early last year. We started being fascinated with the world of censorship and of censorship evasion and how these systems work. And as engineers, we started kind of joking with each other about how we would reverse engineer these things and how we could kind of detect censorship in a way that has never been detected before. So as journalists, we then sort of looked to see what the scene was and started talking to some sources and tried to understand how what work was being done. And naturally, all of our fun ideas had already been tried. There is a vibrant and robust censorship community here in the U.S. And around the world. Harvard has some folks, University of Toronto has some folks doing really remarkable work. And then our story shifted, I think, ultimately to kind of talking about that work and trying to explain how censorship works to an audience of Americans who are thankfully not censored nearly in the same ways. So, and it, as we were kind of having that conversation, we had a very serendipitous conversation with a student of Mark Hansen's about the work that folks were doing here, specifically about Weibo. So I think it's going to be a different show of hands. How many people here know what Sina Weibo is? Many more people. That's great. So Sina Weibo is for ethnocentric Americans like me, China's Twitter. It is a microblogging service that has many, many users, about as many as America's Twitter does. 140 characters, same strictures as American Twitter, except in Chinese. Each ideogram is a word, so it becomes much more like 140 words, and has become a very fascinating and important microblogging service for Chinese citizens to talk to each other, for dissidents to compare notes, and for all sorts of things. But Sina, like all other large media companies in China, needs to self-censor, by which I mean there are staff people at Sina and at other large media companies who function like editors like me, but instead of fixing copy and making everybody's prose better, they delete stories. So if a story runs afoul of a new regime goal, they will delete all of those stories. If too many people pay attention to a story, if it gets too many comments, if too many people are sharing it in ways they don't like, or if the story itself just becomes problematic to them. Often this happens with stories about North Korea. Sina will delete it, as will all other large media organizations in China. So the same goes for Sina, the microblogging service. There are algorithms that stop some speech from even getting posted. If you try to post something about Falun Gong or something, you'll get a note when you try to post it on Weibo saying you're not allowed to post about that. But there are a whole host of other things that you can post on Weibo that will make it through because the algorithms can't detect them. One of them is images, which the algorithms are not yet quite good at detecting automatically. So there is a host of human censors who work at Sina who post facto delete things that have been deleted. So Sina runs this sort of knife's edge where they have to, for fear of being shut down by the government, they have to self-censor. And for fear of having all of their users abandon them, they can't censor so much that people just leave. So it therefore becomes a fascinating kind of reverse image of the self-image of the Chinese government, of what Sina at least thinks will keep them on the good side of both their users and of the government. So, and also the nice other thing about the images is that although lots of research has been done, lots of stories have been done, talking about deletion of the words that go onto Weibo, not a lot has been done about images. And the nice thing about images is that our audience, which is largely here in the US, can understand an image a lot faster than they can understand Chinese text, which most of us don't read. So this is the project that we did. It's called China's Memory Hole. And this, we observed Sina we observed Weibo, I should say, for 2 weeks over the summer, collecting all of the posts being made by 100 people that we thought were likely to post censorable things. So this is sort of the opposite of what the academic kind of statistical approach would be, which is to take a random sample. We didn't need to do that because we knew that a deletion was happening. We didn't need to prove that. We chose 100 people. This was actually very much part of the collaboration with Mark and the team here at Columbia. This was a list that I think the Columbia folks chose first, but it's folks like lawyers, doctors, other people in high-risk occupations who were likely to be posting things that the regime would want to take down. So this is sort of the opposite of a random sample. So we looked for 2 weeks over the summer. We had, I wrote these numbers down, of about 8,000 posts, 524 were removed after the fact by the Sina censors for reasons other than them being pornography. So we have collected them here. We had a team of folks who can read Mandarin, who understand Chinese culture and current events and pop culture references, to be able to not just translate the text, but also to explain kind of the connotations of what's going on in each of the photos. We then kind of classified them into 10 rough groups to talk about, so that people could understand them as groups. A lot of these images are sort of the same image getting deleted a bunch of times. So I just wanted to kind of do a quick demo here. Here is a political speech, and we let you as a reader come and there we go, good. It's all filling in. You can kind of see. So here's a picture, and if you hover, it says the truth, and I can't read this from here, but if you go to the site, it's propublica.org/Weibo, you can see our ultimately a sort of a good guess as to why this kind of thing might be have been censored post facto by the censors, and they fall into rough groups. This guy Bo Xilai is a son of one of the revolutionary leaders who has fallen afoul of the government by, I think, taking bribes and having a wife who I think killed somebody. So he is now in prison, as is his wife, and for the time during which we were observing Weibo computational, with a script, I guess that's computationally, there was a lot of kind of discussion of him on Weibo that was being removed. So again, it was a fascinating collaboration here with the folks here at Columbia. We kind of both have our own mishigas, the academics and the journalists, so it was, I think, a refreshingly sort of a loosely coupled collaboration where we kind of had our own rules, the folks here had their own rules. So Dan Taeyong, who's going to come up in a minute and show Columbia's sort of project that sprouted from the same origin as this, we kind of came to very different places with the very similar data sets. But yeah, that's it. Thanks.
I just quickly want to show the website that Dan has been working on, and the fact was that we had to actually stop our research in order that the ProPublica story could get launched, which we were very happy about, but because we had to, you're welcome, because we had to protect the user's names. So what Dan is showing now is we've stopped collecting. Mm-hmm. We've stopped collecting the data, but we actually collected the data for many months. And we're now starting work on another project, which we're not allowed to talk about until it's finished, with a random sample of Weibo users over a 6-year period.
Yeah, sure. Just to go in detail, we kind of took like a little bit of a more macro view because this was collected over, I think, 10 weeks. And on our end, the question was kind of how do you represent censorship visually, or how do you kind of show the frequency or the activity of censorship without kind of going into the in-depth analysis of each post. So instead of using a timeline, which is maybe a little bit more historical or kind of a fixed-in-history method of representation, this is a way in which the vertical kind of path of the graph indicates the kind of repost count, which is roughly analogous to the spread or the reach of each post. So you can see that on this day, there's a flurry of activity, and this post in specific was reposted something like 6,000 times before it was deleted within an hour and 26 minutes. And then some stats. This was done with an engine that used around like 20 separate accounts to kind of harvest and check every account. So some interesting information. The interesting thing about collecting this data is that how do you understand when a post is deleted? You don't. You can only check back for its absence. So you kind of have to continually check back, and so we checked around once a minute. That was kind of the minimum possible resolution. That came around 100,000 checks on each of these 100 people, so about 10 million checks. And out of 35,000 posts, around 943 posts were deleted, which is about 2.7% of all posts. And you're seeing all the deleted posts here. So that's around 480 posts per day, or 20 posts per hour. And that people were posting, and out of those 480 posts per day, 13 posts were deleted per day. Or, in other words, every 2 hours, one post was being deleted. And one of the advantages of this representation that we wanted to show is that each of these posts is a deleted post, and in some sense, they're short enough to be deleted within 1 minute and 28 seconds. So if you're on Weibo and you're not seeing, there's this universe of kind of these little fragments of deleted posts that you're not able to view just because they're deleted within a minute, within 5 minutes. And so this is kind of the way in which we wanted to represent the activity of censorship, really. What you may see is, you may see longer posts if you check here to there. But in the background, there's kind of this constellation of small posts that get deleted really quickly. And again, as Laura mentioned, we're going to be kind of expanding this research and working with an organization to analyze, instead of 10 weeks, years of, I guess, collected information. Thanks.
So next person is Alex Tyrell from Thomson Reuters.
I think we're one slide ahead. Can we back up one real quick? I guess I could do it. Okay. Thanks, Laura. Real quick, I just want to introduce myself, sort of what I do for Thomson Reuters, a little bit about Thomson Reuters, and hopefully set the stage for, what Laura and I have been working on collectively for the last year to sort of culminate in sort of a really interesting way to use data visualization and something that's, sort of really unique to Thomson Reuters, and was just an amazing experience to work with you on. So a little bit about myself, PhD in computer vision, undergrad in geophysics, so I have a broad range of sort of experiences, multidisciplinary, which I think is, really important for doing, sort of innovation. I'm currently a product developer slash data scientist, and I work in our financial risk division at Thomson Reuters. That's not my keyboard. Oh, down here. Okay. So, real quick, a little bit about Thomson Reuters. So, Thomson Reuters is probably best known for its Reuters news. Okay. So, obviously, Reuters' news is a global, basically. Operating in over 100 countries, 6,000 employees globally. Reuters News employs 2,000 to 3,000 journalists covering global news and events in real time. Fundamentally, we're broken down into sort of 4 different market segments. So financial and risk, legal, tax and accounting, and IP and science. I work in the financial and risk division where we provide information to traders and hedge fund managers and investment bankers, et cetera. A fun fact about legal for anybody who watches Law and Order, and if you see, everybody sort of in that library with all the law books behind them, that's Thomson Reuters. We publish those. Tax and accounting is sort of similar in many respects to the legal business, and then IP and science is actually, a really interesting business unit as well. It's something that typically, universities can relate to. We offer something called Web of Science and Web of Knowledge, which is in many university libraries. We cover massive amounts of scholarly work in journals, et cetera. And, ultimately, Thomson Reuters is effectively the world's leading source of intelligent information for businesses and professionals. That means, we do for the consumer when they're on the internet and they're using Google and various other tools, we do that for the professional who really relies on our content, our information to make business decisions. So, about a year ago, when Laura and I first met to sort of embark on this mission, our focus was really sort of on, the big data theme, which is, very popular right now, and incorporating data visualization and really getting value out of our content beyond what we're able to do currently. And effectively, we sort of focus on sort of some very fundamental themes when it comes to our content, our information, and how we make information intelligent. And as we organize it, we make it very easy to find things, and then we really want to enable people to discover and really explore and find those critical insights. So how do we do this? First of all, we spend a lot of time as an organization connecting the dots in our content, finding all the connections, curating all the information, bringing lots of data together, and really effectively, rationalizing big data. So really putting structure and information. So it's not just a bunch of collections of documents and a large corpus of legal texts. It's really connected, and we really understand what it is. And then basically we organize it in ways that are, really easy to sort of access and sort of, define, and we call it sort of the language of content, okay? And, the idea of mastering is really about, creating this sort of order on the data and doing it once, doing it really well, and letting everybody benefit, and then essentially setting up things that really allow us to leverage big data and linked and open data. And it serves as sort of the frameworks for things like cloud computing, which we're very active in that area as well. And ultimately, it's the single source, the one single truth in our content sets, which is really important as well. So, master the language of content. I like that phrase because one of the cool things we were able to do last year with our financial services product is enable sort of a natural language search. Okay, so in this example, you want to find, price and revenues of Exxon, Petrobras, and Shell in a certain date range. You type it into our Icon desktop product, and it instantly renders everything that you need to know. All the information is there. It's all, it's essentially visualized in terms of charts, but the cool thing is our system understood exactly what you were asking for using semantic technologies, machine learning, and natural language processing. But the cool thing is it knew how to display itself. The data knew what to do once you've asked for it. So we've given you the appropriate charts, the date ranges, we've formatted things, we've colored them. All the tables are laid out automatically. In a sense, we had to go back to the drawing board and say, okay, not only do we need to know what our data is, but our data needs to know what it is. Our data needs to know how to sort of render itself and make itself available. So we really started to put a lot of effort into doing this well, and that sort of enabled all kinds of amazing opportunities for rich visualizations in going more to sort of that discovery phase of finding insights and finding information that you need to make decisions if you're sort of an investment professional, et cetera. And another example of the way we've been able to really leverage data visualization is with our interactive map product, where we're bringing together lots of information about shipping lanes, global supply chain, weather, ports, regions, different types of products like oil and crude and refined gasolines, and it's really on a global scale, and it's updating all the time. And in addition, we have an amazing wealth of information from our news sources that are covering major events like piracy and any sort of disruptions at refineries and things like that would be important. And then ultimately, it's really in a very nice and intuitive, visual way of accessing the information and bringing it all together. So in fact, this piece actually is sort of, the starting point, I think, where Laura and I started to embark on this, way of creating a whole new way of visualizing and understanding our data. And in the meantime, while we did that, at Thomson Reuters, my group in particular, which was relatively small, got a lot of really amazing ideas from Laura's team. And we went back into our group and put our heads together and said, I think we could actually create some pretty cool products around this stuff. I mean, definitely ways that we can sort of bring in elements of design, massive amounts of our content, put it into a visual representation, which although not complex, is actually a good thing because it's amazingly intuitive. So what we actually see here is a product that we created that really allows investment professionals to take like a very high level view of world economies using macroeconomic indicators like GDP and inflation, and really quickly assess investment opportunities using a visual tool and certain workflow components, and also look at the data in various ways. So it's not just a map, it's not just a chart, it's not just a time series, and really find those insights that really sort of were not available in an easy way before. A typical approach might be download a lot of stuff to Excel and chart it yourself. Another approach might be to go to a country guide or page or look at a particular economic time series, and the information just wasn't easily accessible in one unified way in an intuitive interface, which we solved. So, Columbia and Thomson Reuters. This has been an amazing year. So there's just a quick moment here. Laura and I worked very close together. I mean, the initial intention was, really create a visual experience to bring our data to life, which I think we did, which was really fun. We want to make it relevant to customers in financial markets. I mean, that was a goal, and we picked a certain. We picked a certain set of data in a certain workflows and insights that we want to get that were really well suited for that. And then we ultimately want to exchange and share ideas because it's really about learning. It's really about seeing all the amazing stuff that's happening here and getting an idea for, you know, what's on our horizon, and then ultimately do something cool. And I think we definitely did that. It was a lot of fun. Okay, so did we want to show a video right at the end here? Okay, all right. Are you able to, that's not connected to anything. I just use that as a reference. Okay.
Okay, so this video just shows 1 year of shipping of oil-related products. So petroleum and all its derivatives. And it starts very slowly and starts speeding up, showing the whole year, 2012. And the idea here was to basically show the volume, because one of the things we discovered using this data set that Reuters provided was that shipping is mainly an invisible activity, but the volume that actually exists is incredible. So, for example, here, we're only at January 10th, and we already have more than 2,000 shipments of oil-related products, only to and from U.S. ports.
So the other thing we've done is we've created a D3 product which actually shows stories in relation to the macro view of the global shipping. Okay, so the last person is Dennis Tenen, and he's from the English department here, and we're together actually collaborating on a large project with the Columbia Libraries, and we're not going to show that. It's not ready for prime time yet, but Dennis is going to show what we're going to work on together this year.
Right, yeah. And my name is Dennis Tenen. I'm on the faculty of the English department here, and I study book piracy. So I'm going to begin I would like to introduce my work a little bit and also try to I cannot help but integrate a little bit all these great talks that happen here. So it seems, as I was sitting there, it seems that a lot of us are interested in self-organizing stable structures. So whether they're neurons or chairs that you can sit on, or bacteria, wrinkles, or hair, right? Or hairdos, right? These are all kind of some sort of self-organizing stable structures. Brains, perhaps, if you are quoting Minsky, brains perhaps are such structures. And the mystery is always, how do they do it? how is it possible that a brain, right, can develop from a mass, a tangled mass of neurons? Now, the messy structures, messy, stable, self-organizing structures that I study are social. And in particular, I look in the ways specific grassroots communities set up libraries that rival any large library, any large digital library in the world. This is a snapshot of one such community. Now, as you can understand, piracy is a relatively secretive thing. So these folks and people who both share information and people who receive information are reluctant to share the details of their activities. So our approach so far has been to study the infrastructure, the servers, the software that supports book and article sharing activity, and also study to try to get to the social dynamics behind this activity. So this represents a book forum where people collect specific books, mostly scientific material, mostly probably anything that you've ever written or published is part of this particular library. This library is second only to Google Books in size and scope, and I think it has a very important impact on the world, on the way we've collected hundreds of testimonials of people who say that in my country, without this particular resource, I would not be able to do my research. My university would not be able to do my research. So our approach has been to start with the stuff that we can get at, which is online forums, torrent trackers, and then we would also like to, going forward, would like to do field work and interviews. So it's kind of a mix between computational approaches to the study of this phenomenon and field work, historical and cultural contextualization of this work. So where are we going with this? Well, let me show you. Actually, let me show you another one. I'm not sure what that box. Now, these are not published visualizations. These were done. These are more. Think of these as experiments exploring the data set that we have. Here we have collected. This heat map represents specific topics. So here's the humanities, here's the comics, here the warfare, and so on. These are book topics, and we have pretty nice longitudinal data. So we can see the popularity of particular topics over time. You can see over there we have a bi-quarterly quarterly data over there. So where are we going with this? This particular community has managed to create the largest library in the world on an annual operating budget of $1,900. You can kind of compare it to the more official channels of communication like Thomson Reuters, and think about kind of the disparity of access and resources there. How is it possible? How are they able to self-organize and to create stable structures like this? How is this library, how is this particular library, how does it impact our own community, our own libraries, our own publication, the way we publish, the way we access research? So that's kind of an obvious intersection there. Now, another thing that struck me is that Mark Hansen talked about the common challenges to this type of research. The one thing is that and I think it cropped up in several of the talks, is that in collecting data indiscriminately, we end up working with truly large data sets. This particular torrent network that we're studying, we have over 1 million of individual interactions that need to be analyzed. We're now approaching data scales which cannot be done locally. We have to work on a shared computer, on a server. The second thing, the second challenge that we face is that we have a lot of these kind of intermediary objects of scholarship, right? So you have, whether it's a 3D printing model or visualization, it is not clear kind of how we ourselves produce and share such artifacts. We'll call them intermediary artifacts of scholarly production. And finally, I think the challenge that we all share is literacy. Is this a good visualization? How do we read a visualization like this? Are we teaching our students to produce and to read statistics, to read visual information? And that's something that we are trying to, I think, address it altogether. Thank you. That's it.
So, yeah, I know we've gone a little bit long. We had a lot of presentations, but we'd like to invite all of the presenters to come up to the front of the room. And we're just going to kind of turn it over to you, the audience, to ask questions about general approaches or specific projects. And we'll just try to kind of get a conversation started about data visualization with the idea that this is to be continued. So. Are there any questions?
Thank you all for really interesting stuff. Across all these disciplines, I mean, the thing that we initially know unites all of it is the project itself. But I'm wondering if in the process of working through the project, if any of you have noticed sort of shared nomenclatures or like languages that or ways of operating that are starting to cut across the disciplines in terms of how you communicate the data or translate it into other forms. I mean, beyond just simple statistical analysis, but sort of, you know, what tools have you found have allowed you to communicate across fields, I guess?
Well, just speaking from the neuroscience community, dimensionality reduction is a big theme. So, and it's something that, you know, may be going on in a lot of things here. And, you know, and that's kind of what data visualization is. It's how do you compress data? How do you extract out its most fundamental features? And to some extent, that's taking these many dimensions of all of this stuff that's going on and extracting out. You know, you mentioned statistical techniques like principal component analysis, things like covariance, you know, but in general, all of this seems to be kind of a way to represent complexity in a relatively simpler, intuitive way. But yeah, dimensionality reduction is the big thing in neuroscience right now.
But I'd like to add to that just the availability of tools and libraries that allow you to, early on in your research, to explore data. So I would say 10, 20 years ago, this kind of stuff that you saw today would be the end product of a research project. Whereas here, we often, or at least I find it in my work, that we visualize first. We explore just to start forming intuitions about this about the data set. And that's only possible with the availability of the tools and also general literacy, that we have people now, or you know, some training that enables that type of early exploratory research.
Maybe I can add a little something here. I think I agree with what was said, but I think there is another dimension in the question you're asking, and we can all agree with the common denominators of definition, statistics, or mathematics, or the way we display data, and the genres that we can prefer, one with animation, the other one with more static output. But I personally, as a basic scientist, now feel the need of developing a vocabulary or sets of type definitions that can go across boundary, not only in different scientific field or humanities or art, but something that transcends the entire human endeavor. I think there is convergence in the way we think about big data processing, visualization, and others, but I'm not convinced that when two different people from two different fields are talking about very similar display output of a very large data set that are actually using the same kind of language and vocabulary to communicate in the best and most efficient way. There are terminology, for example, in Latvia that comes from 18th century, 19th century, that is still now being used, and different people use it to mean different things, and it's a very dangerous thing to do if it's not fixed relatively soon. So I am hoping for some sort of international standards or agreement, and I think Thomson Reuters is probably a great forum for that, to at least engage in a debate where scientists, people from humanities, art, architecture can agree on the sets of definitions of words from which we can no longer diverge. We need to have common sets of rulers if we're going to measure things and comparatively between different fields. We're no longer allowed to use rulers that have different units on them. So I think this is, to me, one of the biggest priorities as we move forward, and it was a great experience communicating with you guys as architecture students coming to a science lab to figure out how many of the words that we're using that for us are perfectly defined with strict boundaries are kind of fuzzy in the mind of otherwise very smart people. So I think we need some sort of a convergence on that.
But maybe that's the point of data visualization in general, is that it takes it outside of lexicons and puts it into this very primal, you know, I remember like this trading platform, this is back, you know, in the internet boom, that they were going to have this virtual reality trading platform where you would see all the stocks as this surface, and when things were changing in one domain, you could reach out and kind of like manipulate that in a kind of, in a physical way, right? And, you know, making use of like our visual, you know, it is this primal thing. We don't even think about it. It's, you know, when we see these, you know, the dots changing or things speeding up, like, I don't know if you saw like the visualization of the nuclear arms race, all the nuclear tests that had been done in the history or something like that. And, you know, just seeing that, your visual system takes that and transmits it into kind of experience. And, you know, it's very fundamental, and it's outside of like a lexicon, you know, and that's the cool thing about what a lot of you guys are doing, you know.
Isn't that the danger, though? Isn't that the danger, to think that there is not a literacy here, that there is not a rhetoric being deployed, that where there is no common agreement on how to read a visualization, but there are standards and connotations that we can start teasing out if we actually talked about it. If reverting back to a sort of kind of visceral, oh yeah, we get it. That visualization in particular.
Yeah, I mean, I think there is like a facile understanding, but right. You know, I think that maybe, you know, if it's just based on sheer data, the data will set you free to some extent. You know, and maybe there's, you know, it is susceptible to being manipulated in certain ways, and you do have to be careful. And as people get more practiced in this, then, you know, maybe that will become more of a problem. But, you know, that seems like one of the things about big data is that you can trust it just because it's the statistics of all of this information itself kind of shows its order.
Yeah, go ahead. Just as a response to, I mean, I do think that there is a kind of visual language within the representation. I think, you know, making whatever visualization, there is a process of understanding how these things are interpreted. And, you know, you go the spatial world, you have like years, decades of training on how to understand the spatial world. I think the same goes for the visual world, and then all visualizations will be a practice in like how do you navigate both what people understand and how to represent things. For example, right now, like a filled-in box with text inside of it, we can now understand it as a button to press because of, you know, the past, say, decade of web interfaces or whatnot. 10 years ago, perhaps no one would understand it as a button until it had this kind of skeuomorphic, you know, shaded thing. So I think understanding how those things work right now in context to like our day-to-day lives, and understanding what the kind of prevailing different visual languages are, and then fitting visualizations within that is maybe kind of a task to do.
As a journalist, I really respond to this notion, and I love this phrase, the dimensionality reduction. As on some level, I see that as my task as an editor, right? It's the same thing. We just call it editing. But I think that there is a trust that you are asking your readers or your audience to have in you, that you've reduced to the right dimensions, and I think that you have to back that up with a lot of transparency. Again, this is just kind of purely in the journalism world where I have sort of 10% more knowledge of my readers about any given subject, and I need to express to them all of the biases that might have gone into my data selection, all of the other paths I might have taken and why I rejected them, show them all of my work, as much of my raw data as I can. And as a data journalist, we are, I think, required as a matter of our journalistic integrity to expose those things, and we invariably do in the projects we do. But I think you're absolutely right. I think this kind of process of commensuration, or whatever you want to call it, I'm at Columbia, so I can use that word, these are hard things, right? And we're going to get it wrong, and we can talk about it, but I think ultimately the market is going to, the marketplace of ideas and regular people are going to decide what works and what doesn't work, and what they trust and what they don't trust. And I think just the important thing is that we are honest with ourselves and with our readers and with our audience about how we're doing our thing.
First of all, I thought all those were really fascinating. And as a programmer, I wonder, how do you assess the success or failure of a given effort at data visualization? I've always sort of wondered at the responses I get from things, and I found it very easy to wow crowds with or without actual merit to the product. So I wonder how you think about that.
Actually, why don't I jump in real quick on that? That's a really interesting point that you bring up, because we're really trying to do things for our customers that they find really exciting, innovative, but they ultimately have to really add the value, right? I mean, they have to actually help them do something better, faster, or with greater understanding. So while we do get people really excited about some of the things that we've built around data visualization, we've done many different things. At the end of the day, we do have products that we've created that really just open up new doors, right? And we try different things and try to get it right quickly as often as possible. But at the same time, I mean, you have to have a little bit of room to experiment, right? Do something that's really sort of innovative, like the stuff that Laura and I worked on. Like maybe it turns into like a product idea, or maybe we can put this into something that Thompson Reuters is doing in the near future. Maybe it's just ideas, and maybe we take it to some customers, or just iterate on it until we sort of have it refined and can prove it that this is helpful versus something else. But you really bring up a good point, especially as this stuff becomes sort of more commonplace, right? You sort of run into that sort of dilution factor sometimes of just having a lot of data visualization.
I think visualization is kind of a shortcut to analysis, right? And that's the worry, is that what if it's just a shortcut and you didn't do the work? And I think that's why it's so exciting to see the collaborations here, where you have powerful visualizations with really powerful research and science behind it. I think several of the videos that we saw today, or presentations we saw, had this magic moment where the visualization becomes the thing, right? Like the chair, and then somebody picks it up, or whether it's the bacteria, and then it's something you can touch. And I think that's the moment that you hope that happens in every research project, where you're not just talking about models or pictures, that they, in some deep way, represent reality underneath.
Do you feel like you're developing an instinct for, as you're developing the visualization, knowing or sensing that you're maybe going down the right or the wrong path with that? Because I've when I think about this stuff, many times I've tried to visualize something and then I just get a bunch of mush and nothing emerges.
All right, so maybe I can answer that by going back to your previous point first. You ask, how do we know the difference between something that's a big wow, you look at it because it's aesthetically pleasing, versus something that you learn something from? And that's a fundamental question in science. When you teach your graduate students or your postdoc, the tools that you use to teach them the difference between a 3D model or a rotational Z-stack that you saw in a couple of talks versus the reality of what the functionality of this structure is. And I think there is room for both of them. It is true that for science, you really have to end up asking, what did you learn? So you go to a beautiful talk and you see amazing, very sophisticated sets of movies and animations and reconstructions and Z-stacks and you name it. But then when you come out of it and you think about it for 5 minutes, say, is there any new information in that presentation that I didn't know before I walked into that room? Or is it a better way to show something that I already knew? And a lot of the time, because we're aesthetic and visual people, the beauty of the information itself is enough to grab us. But a good test at the end of the day, or at the end of the talk, is to say, okay, how much did I learn for this 1 hour that I spent? And that difference is different from one person to the other, and I think certainly in basic science, which is, you have to remain pretty much Cartesian, what is really important is what you get out of a series of large-scale data analysis. But of course, the more beautiful your information is displayed, the easier it is for the students usually to grasp. It's a combination of the two.
And you actually brought up the instincts, and starting from our data set, we started with our instincts, how should we address them. And what I really benefited from this data visualization project is that we got new ways of presenting the data, and that they completely went against my instincts. So just by looking at this new way of presenting it, we could form new ideas. And of course, at the end, this could all be random and not meaningful, but then that's the power of going back to the actual experiments and testing these new ideas. And I think it's just a new way of opening new horizons. And that's what I really got out of that.
I think we probably only have one more question.
No, I'm just
one
looking for other
yeah.
questions. Yeah.
I think since we're running out of time, let's have one more question and then we'll wrap up.
A lot of pressure on somebody.
So we talked a lot about reduction, right? And also about how sort of a cultural momentum kind of teaches us how to read these things in a particular way that's satisfying to those who are producing them, and of course that allows us to answer those questions at the end of the 1 hour talk. Did I learn something? And some people will have different. There will be degrees of these things. But what about not reduction, but the addition of noise that some of these visualizations bring? So just a simple example is in any given network visualization of social actors, one that I'm familiar with. I come from the Department of English, so I'm familiar with recent visualizations that have appeared about the relationships between characters in Hamlet. And I've already been at 2 talks in which Franco Moretti, the professor who is famous for these visualizations, has had to answer questions. What is the line, what is the distance between the characters in the network visualizations done with Gephi or D3, I think, mean? And of course, they don't mean anything. They were, but some people are quietly interpreting them as meaning something. And this is just one example of the ways in which sometimes visualizations add things that were not there. They add dimensions that were not there. And unless we, of course, record the reaction of everybody in any given room, we don't know what people are taking away from some of these things. So what is the false knowledge that you fear or might be being introduced into the world? Or should we not fear that because it's so minimal that it's negligible?
Well, just speaking from experience with data analysis, that noise often gets smoothed out with a larger data set. And so, I think that, maybe one of the points of doing some of these things with really large data sets is that, you can see what some of the fundamental, coordinate axes, like Cartesian axes of these phenomena are. And yeah, I mean, you will see maybe you see some blips in the Exxon shell, Royal Dutch shell thing that are in like weird places or something. But one thing about like our neural systems is that they're really good at kind of screening out noise. And so making use of our visual system, is kind of, I mean, you're right that you should be worried about, sometimes the tail wags the dog and the outlier might be really super important. But I think that with big data sets and if represented in a reasonable way, it kind of will smooth out or average out that noise to find out some of these fundamental kernels.
I mean, kind of an obvious answer is these tools allow us to see phenomena on a scale that's not visible to the naked eye. So I think, for example, doing network visualizations of Hamlet is not great because I can read it pretty well myself without sophisticated tools, but looking at long-term trends of whether in my pirate network a particular topic all of a sudden becomes popular or some tweets are starting to be repressed, that stuff is nothing you can get just by looking at Twitter or just by reading a single book. So you get kind of the scale there. It's a whole, as you know, from the theory of this, is you get a different scale of analysis, whether it's a macro scale or a micro scale.
I think noise is actually very important for large-scale data analysis because it defines really the significance of the conclusions that you're willing to make. The farther the distance between the signal and the noise, the more significant the quality of the information. So in the absence of noise, it's very difficult for me to say how significant two-fold, three-fold, or 300-fold differences are. But if I have a baseline, even if that baseline fluctuates within given boundaries of dynamic resonance, that helps me figure out how far above or below that regular white noise I find myself. And the more I have this, the more confident I can be about the value of what I'm measuring. And perhaps for the wrong reasons, in basic science, two-fold differences or less are not considered very serious. But if you go 5, 10-fold or above, then suddenly your data has a different kind of strength in terms of significance. Without having the noise, it's very difficult to assess what the importance of what you're measuring is. I personally welcome it in my data. I hate it when the distance is very small. That means that I need to tune up something that I'm not measuring what I'm measuring with the right tools. But I cannot really do without.
I want to thank you all, and I don't think there's ever been such a diverse group of disciplines in the architecture school, and it really is amazing that through data there's so many ways that we can talk to each other and that the work has relevance to each other, and I think that's the thing we really learn from this project, and also what the limits are with all of these things. So thank you. Yeah.
Thanks.
Thank you.